In this session, we are going to discuss typical protein sequence. So, we have seen that the sequences, either a DNA sequence or RNA sequence or protein sequence, are the core topic that a bioinformatician always will discuss. So to see how a protein sequence looks like, and how proteins the different chains present in a protein sequence, the characteristics and other parameters associated with the protein sequence. So we are going to just look at a sample protein sequence, and the question is why we have selected insulin, that to human insulin, as our case study. So as a bioinformatician, maybe if we are not so much sound with biological terms and other wet lab experiments, so even though we must know how a typical protein sequence will be looking like, or what are the properties and characteristics of normal protein sequences. So for this, we take human insulin as our target sequence. Why is that? It is a typical protein which has all the properties of almost all the basic properties of the protein sequences, like the different chains, alpha chain, beta chain, the disulfide bonds, the interactions, etc., are present in this human insulin sequence. So as we know, insulin is a hormone, and it is produced normally in the cells of pancreas. And so as a hormone, it is essential for regulating energy storage and glucose metabolism in the body. So there are two functionalities of insulin as a hormone. One is its energy storage, regulating the energy storage, and glucose metabolism. So human insulin protein is actually insulin in liver or muscle or other fat tissues. It actually for stimulating the cell to take up the glucose from blood and store it as a glycogen in liver and muscle. That is why we say it is a regulating energy. It is essential for regulating energy storage. So as we know, failure of insulin control causes diabetes disease, and insulin is actually it is a small protein. And that is also another reason why this human insulin is taken as a case study for understanding the features of a typical protein sequence. So it is composed of actually 51 amino acid, or you can say that human insulin sequence has a length of 51 amino acids. So as we know, the molecular mass of human insulin is 5808 Dalton, and it is released by the cells of pancreas, and it is actually it has two chains, A chain and B chain. So they are linked together by disulfide bonds, that is sulfur to sulfur bond. Insulin structure actually varies slightly between species of different animals, or we can say that, or in other sense, we can say that the insulin structure is almost conserved between different species. And it is encoded by a particular gene called INS gene. Actually, this gene is located in 11th chromosome. So as we know, each gene, each protein is produced by a particular gene, which whose location will be studied in advance also, which can be studied in advance. And as far as human insulin is concerned, it is or any type of insulin, it is human insulin is concerned, it is present in the 11th chromosome, and at position say 15.5. So this is the position of the particular gene, and it is always taken as or considered as a small globular protein. As far as the structure of insulin sequence is found, is observed, it is actually first characterized in long way back, say 70 years or so, in 1951 by Frederick Sanger. And normal insulin that is biologically active is monomeric or exists as a single molecule normally. And as we have seen, it has two long amino acid chains, alpha chain and beta chain, or you call it as polypeptide chains. So the A chain and B chain, so put together we have 51 amino acid, and the molecular mass is 5808 Da, and it is a heterodimer, of with A chain and B chain. And so these chains are linked together by, as we was seen, by disulfide bonds. So we can see the chemical formula of human insulin over here. See, carbon atom, 257 carbon atoms, 383 hydrogen atoms, 65 nitrogen atoms, 77 oxygen atoms, and only 6 sulfur atoms. So all these 6 sulfur atoms are involved in disulfide bond. That is very curious factor we can observe over here. So there are 6 sulfur atoms, and they are participating in 3 disulfide bonds. So the two disulfide bond, as we can see in this figure, it is between the two disulfide bonds are between A chain and B chain. So say this is A chain and this is B chain. There is one, here is one sulfide disulfide bond, and another one. So this connecting the A7 residue to B7 residue, and another one connecting A20 amino acid to the 19th amino acid in the B chain. So there is another disulfide bond, and you have an intra disulfide bond in A chain itself. So so altogether we have 6 sulfur atoms present in human insulin, and all 6 are involved in forming disulfide bond. This A chains and B chain, A chain and B chain are connected together by 2 double sulfide bond, and put together they will be put together by these 2 disulfide bonds. And the internal disulfide bond, actually it is between 6th amino acid and 11th amino acid. So between A6 and A11 there is an internal disulfide bond in the A chain. And here you can see the amino acid sequence as we have seen. A chain consists of 21 amino acid. So A consists of 21 amino acid, and B chain consists of 30 amino acids. So put together we have 51 amino acid which form the human insulin sequence. We can see the amino acid sequence over here. You can see the two double disulfide bond connecting A chain and B chain and putting them together, and another internal disulfide bond in the A chain also. And this is the 3D structure of human insulin. And here also magenta color represents the disulfide bonds present in human insulin. So as we have seen over here, and studying about a protein sequence, to start with a sequence, you can take this amino protein sequence, human insulin protein sequence, because it is very small and it was only 51 amino acid, and the simple two chains, A chain and B chain, but it has almost all properties of like any other complicated sequence, protein sequence. It is a heterodimer, and it has two chains, and it has alpha helix and beta sheet, beta turn, and it has high order assembly. So all the structural features of a typical protein is present in human insulin protein. So and on the at the same time, it is very simple, it is very small also. So to start with a protein sequence, we will always take this as an example.